Okay, colleagues, please take a seat. We are going to start the second session of today. This is more focusing on methodological issues, and Dr. Blumenthal from FDA, perspectives on non-randomized trials in oncology drug development.
Okay, thank you, Dr. Pignati, Dr. Casali, for inviting me. This is a very timely and important meeting. Issues that we struggle with across the pond at the FDA. How do we manage uncertainty? How do we define what thresholds for breakthrough therapy designation, accelerated approval? When do we need randomized controlled studies? So this is sort of a highly reductionist jelly bean plot outlining challenges with what I term the old paradigm. My field, what I focus on is lung cancer. So this is just an example where you're taking a molecularly heterogeneous patient population with known and unknown oncogenic drivers or tumor suppressor gene mutations. Sometimes you don't know what is a driver. And then within a randomized controlled study, enrolling, say, 1,000 patients to, in the frontline setting, for example, platinum doublet versus platinum doublet plus drug X. And the risk you run here, and as we've seen in the last couple decades in oncology drug development with so-called failed phase 3 studies, is you run a high risk of your study not meeting your statistical endpoint, or as has been discussed this morning, you might detect a clinically small, some may say trivial effect. In the new paradigm, using, say, ROS1, as we discussed this morning, as the poster child, you identify your oncogene, you give them a targeted therapy, study it in a small patient population, and you see a large magnitude of durable response rate. And the challenges here are, one, what's the feasibility of screening these patients? One could argue ROS1, 1% of a very common tumor, 2,000 patients. We do randomized controlled trials in mesothelioma, but those patients can be phenotypically identified. ROS1, at this state of, in this day and age, without universal genomic screening, very hard to identify those patients, screened and enrolled in a randomized controlled trial. We're going to be seeing this more and more with the increased uptake of genomic screening with the long tail of these rare and infrequent genomic drivers, finding the proverbial needle in the haystack. The second question I'll allude to in the next slide is, do we have equipoise to even conduct these studies when you see such a large effect in early clinical development? So this really came to the fore once again with the development of vemurafenib, a BRAF inhibitor in melanoma. There was a New York Times article which highlighted it very well. They actually profiled two cousins. One was randomized to the experimental BRAF inhibitor, the other to a toxic and marginally effective chemotherapy agent, dacarbazine. And I won't read the quote, but they questioned whether, with the new science, whether it's ethical to conduct a randomized control study. And so more and more there was discussion of clinical equipoise, the assumption that there's no one better intervention present during the design of a randomized control study. And when there is no clinical equipoise, should a randomized control study be conducted? And then oftentimes we get into these quandaries. We don't know exactly, no one really knows who's really requiring these randomized control trials. We'll get on the phone with our health authority colleagues every month, and it's not clear. Are these regulators? Are these payers? Is it drug sponsors that want a competitive advantage? Maybe they want a survival advantage over their compatriots. So these are some of the quandaries that we face every day. You've seen this parachute test numerous times, so I won't belabor the point, but throughout medicine, when we see unprecedented effects versus historic control, we don't need a randomized controlled trial. One example that hasn't been brought up yet was multi-agent chemotherapy for testicular cancer, 70% complete response rate, cure rate models versus a contemporaneous control. That was enough to satisfy both regulators, payers, the field, that obviously this is an effective regimen. Of course, we also have cautionary tales. One would be autologous stem cell transplant for metastatic breast cancer, adopted early in the U.S., and then the randomized control studies failed to show a survival benefit. So our stance at the FDA is we use objective response rates in single-arm trials for regulatory approval of large magnitude and long duration. Why response rate and not time to event endpoints? So we know that tumor regression is directly attributable to the drug. Spontaneous remission is rare to non-existent. So these are controlled studies. Our historical control is, our null hypothesis is no response. We don't use time to event endpoints, prolonged stable disease, progression-free survival, overall survival in a single-arm study, because this could be due to an indolent tumor type, the natural history of the disease, and not the intervention itself. We published a paper. This was a multi-stakeholder initiative. I would direct you to the publication in Clinical Pharmacology and Therapeutics on what would be some of the criteria in which we'd use non-randomized studies in the evaluation of oncology drugs, strong scientific rationale, preclinical data, well-defined patient population, benefits outweighing the risks, randomization unethical or unfeasible. This is a partial list of accelerated approvals based on response rates in single-arm trials. This is a well-defined mechanism at the FDA dating back to 2001 with imatinib. Many of these are new molecular entities, so the first approval. Some of these are efficacy supplements. I would point out that most of these drugs, imatinib, brentuximab, crizotinib, osimertinib, pembrolizumab, ibrutinib, these are sort of the breakthrough type drugs, the transformative type therapies that have gone on, many of them in randomized control studies, to confirm clinical benefit. And in some cases, we've also granted regular or traditional approval based on overall response rate in single-arm trials. Again, this is a partial list. Some of these are efficacy supplements, as alluded to this morning with crizotinib and ROS1. You already had a well-defined safety and efficacy database in the ALK setting, so it wasn't a very high risk to a leap of faith to approve it for ROS1. Some of these are, in fact, full approvals based on single-arm trials. In most cases, these have been in cutaneous lymphomas or basal cell carcinomas, where you have these fungating, disfiguring lesions, and inducing regressions in these tumor types was thought to be evidence of direct clinical benefit, improved cosmesis, and thought to be very meaningful to patients. So those are the types of scenarios where we've done full approvals for new molecular entities based on single-arm trials. We've also done sort of surrogacy analyses. This is one that we did in lung cancer, 14 trials, over 14,000 lives. And what we found was that response rate was strongly associated with progression-free survival, as you can see on the left on a trial level. The middle panels actually show that response rate doesn't seem on a trial level to correlate with overall survival, nor does PFS. However, there are caveats there, including high rates of crossover, long post-progression survival, which may not enable detection of an association. And then, of course, on a patient level, we know that patients who respond do better than patients who don't respond, even if you do sensitivity analyses like Landmark, excluding those rapid progressors. patients who respond tend to do better than non-responders. We also, in the morning session, we talked about how do we tease out predictive versus prognostic. This is one example of academic investigators, including Alice Shaw from the Mass General, looking at ALK. These are small numbers, but what they did was they went back and looked at their archived tumors databases, looked at clinical outcomes, and they saw that patients who were ALK positive, treated with conventional therapy, did about the same as those who were so-called wild-type patients treated with chemotherapy, and those who were ALK positive treated with crizotinib did better. And their conclusions was that ALK is not a favorable prognostic marker. I think in this era of big data, we need large, high-quality registries to provide useful prognostic information. One database that we've seen drug companies use is the French lung cancer registry, in which they're prospectively testing patients for a number of driver oncogenes. And we've actually seen that they have been able to discern prognostic information from these data sets. So I think it's very encouraging and hopefully signs of more progress to come. So why are we uncomfortable with single-arm trials? There are a myriad of reasons. One is cherry-picking the patients, your marathon runners, your best patients. We know that randomization addresses known and unknown biases. There's a fear of dialing up the dose to get a response, but perhaps compromising long-term toxicity. There's no comparative safety data, and you don't have any data or limited data on prognosis in your biomarker-defined subsets. We feel that randomized control studies, sort of the gold standard, if you will, of evidence generation, will still be for the majority of cases, particularly if you have a cytostatic drug or an immunologic mechanism of action. You may need that randomized control study to assess benefit risk. Also, if you have poor understanding of the toxicity profile, lack of understanding of the natural history, when you don't optimize the dose or the biomarker strategy, randomization will be very helpful. And of course, in settings where there's a vast array of effective available therapies, then the bar rises to show that you're better than, say, the standard of care. These are some recent challenges we've had in the field of single-arm trials. The first was ruxolitinib, which is a third-generation EGFR T790M tyrosine kinase inhibitors. We actually took this to the Oncologic Drug Advisory Committee just a few months ago, and we discussed a number of uncertainties. One was surrounding the dose of the drug. Another was the efficacy, the activity of the drug. For accelerated approval in the U.S., you need to be reasonably likely to predict clinical benefit over available therapies. And there was intensive discussion of whether the response rate and the durability was better than available therapies. And finally, there were concerns about the safety risk, particularly QTc prolongation leading to torsade de pointe and potential sudden cardiac death, particularly in patients who were slow NAT2 acetylators. And ultimately, the Oncology Drug Advisory Committee voted 12-1 to recommend postponing approval pending the results of a randomized control study. Subsequently, after that, the company actually announced discontinuation of development of ruxolitinib. Another just challenging, and I don't mean to single out these agents. I just wanted to, because as was alluded to earlier, we only sometimes talk about success stories, so sometimes it's also good to talk about challenges as well. Ponatinib, a third-generation tyrosine kinase inhibitor for CML and Philadelphia-positive ALL, including the T315I mutation. This drug is marketed. It did receive accelerated approval in 2012 based on single-arm trials, but perhaps the safety profile may have been underestimated in the initial database. In October, of the following year, the sponsor voluntarily suspended temporarily marketing of the drug because of increased cardiovascular events, including vascular and arterial occlusions, and subsequently resumed marketing with risk mitigation plans and as well as modifications to the indication and to treat a more refractory patient population. And I think we'll also potentially talk about alternative designs. You don't have to do a single-arm study. There are adaptive randomization designs like the I-SPY2 study. There are randomized dose-finding trials. I think Dr. Rubin in the Keynote-1 study talked about actually within that large phase 1 experience, there was randomized to dose and sequence. Also in this era of electronic medical records, perhaps we could do more pragmatic real-world randomized controlled studies. And finally, something that we've talked a lot about in the agency are these master protocols testing multiple novel agents against a standard of care in which the novel agents wouldn't necessarily be directly compared, but just comparing to the control. So perhaps there's some parsimony there in a shared control arm. We haven't seen a lot of enthusiasm on the part of the industry to embark on these trials, but we still think there could be usefulness to this approach. This slide is just to say, and we discussed it somewhat this morning, responses may be qualitatively or quantitatively different, but still be deemed partial response by RESIST. On the left, you see a response that's quite obvious, seen from across the room. On the right is a response where in the journal it required an arrow to point it out. So there are efforts to assess different metrics of response, including overall tumor burden, volume, depth of response, durability of response. I would say from a regulatory standpoint and perhaps from a clinical standpoint, if we go changing the RESIST criteria too much, it may be hard to compare to historical benchmarks, which are important for us for both breakthrough therapy designation, accelerated approval. So there is some caution to be sort of tweaking the gold standard. Nevertheless, we do think that this is important to look at, and there's a multi-stakeholder effort within the Foundation for the NIH to look at, it's called the VOLPACT initiative, to actually look at radiographs from completed phase 3 trials and assess different metrics of response. And hopefully they'll also be getting immunotherapy trials as well into this VOLPACT initiative. So just some parting thoughts. Response rate of large magnitude and durability in single-arm trial is an established mechanism for approval, both accelerated and in some cases traditional approval in oncology. Many of the approvals based on response rate have been sort of these breakthrough-like therapies and have been transformative for patients. There are some limitations, as was discussed, optimization of dose and biomarker and assessing safety. Big data, data sharing, and master protocols may be necessary to pool resources, drive down the cost of these trials, and to study and enroll rare and underserved subsets of patients. And we talked about the alternative trial designs, alternative metrics for response that are needed. And then finally, I think we also need sort of, if possible, a global discussion between the regulatory bodies, the payers, the national research institutes to try to decrease the number of randomized controlled studies that are either uninteresting for patients and clinicians and where equipoise is lacking. So thank you.
Okay, I guess that we should go to the next presentation straight ahead, and then we have the discussion in any case at the end of the session before lunch. So Paolo Bruzzi from Genova, Italy. Can we improve the reliability single-arm trials? in
Well, let me start saying that I agree on the criteria Dr. Blumenthal presented for non-randomized single-arm trials, and I will not discuss any further that issue. I will focus on experimental studies aimed at evaluating the efficacy of with these phase 3 trials. I will not discuss phase 1, 2, those finding proof of principle studies or observational studies of cancer drugs. Will not discuss other treatments or other disease without a randomized control group. And we have two options for single-arm trials without a control group, and we saw this morning 2 examples of this, and with one or more control groups, and we have examples. Most of the examples I will present come from old studies, because I think we have to sort of remember what were the methodological issues that were discussed in the past decades as far as single-arm trials are concerned. Without a control group, you usually look for some sort of success rate, proportion of the responses of long-term survivors. And this, of course, is fine when you have breakthrough drugs, but we will not discuss any further breakthrough drugs. But otherwise, success rates are very unreliable because we see large variations. We have been used to large variation, huge variations in the response rates to anthracyclines in breast cancer, any sort of chemotherapy in solid tumors. Even long-term survivors, it would have been difficult to interpret the results with ipilimumab if we had not had the control group which shows some sort of long-term survivors with dacarbazine alone, which could have been guessed based on old studies that showed that there is always a proportion of long-term survivors in metastatic melanoma. But if instead of success rates, we use some more commonly used statistics such as median survival, again in metastatic melanoma, we can see twofold variations even with the same treatments. So it's very risky to have not to have a control group. But I would like to make an example. We discussed already last year in this room of a trial by Silvia Stacchiotti. This was a trial in a very rare tumor, chordoma, 56 patients, no control group, and 1 response out of 50 patients. And due to the lack of a control group, it was not possible to assess the meaning of the 9 months median PFS and 35 months of median overall survival, which led to the conclusion, to a negative conclusion, while indeed this trial could have been promising in terms of stabilization of the disease had we had a control group. So my first statement is that a control group, with the exception of breakthrough therapies, a control group should be always available for comparison and considered in study design. So we have 2 types of control groups, historical and concurrent. And the idea is to build up a study design resembling a randomized trial, where instead of random assignment, you have a sort of assignment due to period, center, doctor, patient. And we have 2 main problems with this design: unbiased assessment of the outcome. And we know that if we have a hard endpoint, we don't have this problem, but we have other problems due to crossover, to confounding for following therapies. But with soft endpoints, we should always be aware that there are problems of bias beside the validity of these surrogates, of these endpoints as surrogates of overall survival. Can we make sure that their assessment was unbiased and comparable in different periods of centers? And I would like to, it was already mentioned, I'd like to mention that progression-free survival is probably one of the softest endpoints because of its dependence on the intensity of follow-up checks and on the quality type of follow-up checks. And blind review of endpoints is not always feasible, and sometimes it's not effective because the types of documentation of records we have from the historical controls is so different. And again, if possible, the study endpoint should be overall survival. Otherwise, a bias must be always suspected and considered in the interpretation of the results. Then we have a second problem. How can we make sure that the two groups are comparable? And the problems with historical controls are many. We have been discussed for decades. But I would like to make the point that statistical adjustments do not work to remove this bias, especially stage migration, the well-known Will Rogers phenomenon, is not adjusted away by multivariate analysis or propensity score analysis. And these are data from our cancer registry in Genoa. We see an improvement in diagnosis, in increasing the number of cases diagnosis over time, but the distribution of stages over time does not change despite earlier and earlier diagnosis. This means that there has been a strong Will Rogers phenomenon, a strong stage migration. Patients are staged better and better over time, and which leads to intra-stage improvement in survival, even in stage 1, where nothing has changed during the last 40 years. Stage 2 and 3, much in excess of what you would expect based on adjuvant therapy, and much less in stage 4. But even in unknown stage, you see an improvement. So basically, this is another example I'd like to make. 2 consecutive trials with the same, almost the same control group, same therapy, same selection criteria, largely the same centers. And you see, with 5 years difference, a hazard ratio between the first control group and the second control group of 0.73, which means a 27% risk reduction, which is astonishing for anyone who's been involved in adjuvant therapy of early breast cancer. It's much more than you would expect by adding a drug to an effective drug to the adjuvant regimen. And it was statistically significant. And the third advice is that the historical trends in survival must be accounted for, which would suggest concurrent controls, but we have many problems. Where do we find concurrent controls within the same centers? And this poses ethical problems, possibly in other centers. How can we impose to the controls diagnostic and molecular or staging procedures? Do we need an informed consent from these concurrent controls? Do we have problems in very rare diseases of sample size? And we have 2 options, natural cluster experiment or registries. A natural cluster experiment is when we have centers where the new drug is introduced during the trials and centers where the new drug is not introduced. And we had an example, it was not really a new drug. It was a sort of retrospective analysis, but as an example of this kind of experiment is adrenocortical carcinoma. The study was made by 8 tertiary referral centers, 4 currently using adjuvant mitoten and 4 never using it. And we had a second control group that included all cases included in the German registry where adjuvant mitoten was never used. And the 2 control groups differed to some extent, but the mitoten group stood out as clearly showing a better prognosis. So this was seen as a demonstration of efficacy of adjuvant therapy. When it comes to registries, we have 3 types of registries. The population-based registries, which are not very useful because they are representative series of patients. They can be used to assess historical trends, but the quality of the clinical data is so poor that they cannot be used. You have institutional-based that suffer from small sample size and from selection. And then you have sort of disease registries based on one disease, on many institutions that collect data on one disease. And they have the same advantages, good clinical and molecular data of single institution registries. And the selection bias may be present, but often there is a satisfactory population coverage, especially if there is, we are dealing with a rare disease. For instance, in the Italian Neuroblastoma Registry, we had a coverage of more than 80% of all incident cases in Italy of neuroblastoma. And follow-up was homogeneous, and we had homogeneous treatment protocols. And we were able to show that intensification of chemotherapy or adding new drugs in protocol B and C, this was a 12-year study, 3 consecutive protocols, failed to translate in any improvement and only was not associated with significant improvement in 5-year survival and only increased the toxicity. As a consequence, prospectively planned comparison with appropriate registry data could be very useful. And we might conceive a sort of 2 types of prospective of studies of single-arm trials. True prospective core studies, where the comparison is between center participating, not participating to the trial of a new drug, or single-arm trials nested into population or disease registry, when the drug is introduced into a group of patients selected of a registry of incident cases. If it's possible, you have a concurrent control group of patients with similar characteristics, and you can estimate the historical trend in prognosis, and you expect a sudden change in prognosis only in patients eligible for the new drug. If these are 3 stages or 3 molecular variants of the same disease, and these are the increase due to the historical trends, when you introduce a new drug in a specific subgroup, you expect only patients receiving the new drug to show a benefit along the same historical trends. The requirements, of course, would be a blinded review of selection criteria and prognostic factors, but the availability of a prospective registry would make sure that all relevant endpoints, including quality of life endpoints, can be collected over time. Analysis should resemble that of a randomized trial, and comparison would be with the expected overall survival based on historical trends and overall survival observed in centers not participating to the trial. Again, we should not rely too much on statistical adjustments such as multivariate analysis or propensity score. The question is whether positive evidence of a study of this kind, from a study of this kind, would be deemed sufficient for registration of a new drug in the presence of not miracle drugs, I mean, not a breakthrough drug. And if yes, I think that establishment of prospective disease registry would be critical for instances when randomized clinical trials are problematic due to grim prognosis and or rarity. But we should make sure, again, that single-arm trials do not become a shortcut to registration. Single-arm trials should have the same requirement in design of a randomized trial, with the only exception of randomization. And for this reason, I agree with what was said by Dr. Rubin. They're even more demanding in terms of the planning, design, statistical planning, and analysis. But we should not forget what we've learned from frequent tumors in terms of potential biases affecting non-randomized trials. Thank you.
The next speaker, Dr. Murphy, for Project DataSphere, Progress and Promise of this Digital Library Laboratory.
Thank you, Professor Pignati and Professor Casali. It's a great honor for me on behalf of the CEO Roundtable on Cancer and one of its products, one of its proud products, namely Project DataSphere, to give you a progress report. As I was looking at this very splendid room, thinking about the architect who designed it, it must have been a Norseman or someone from Norway, because it's built in the form of a Viking ship. And I feel as an oarsman now, actually. We've had such great speakers who have propelled us forward. I'd like to pause to actually hopefully join them by telling you a very interesting story. It is about the origins of this enterprise that is intended, first of all, to be derived from you. It is devoted to you, that is, every one of you, because it's ultimately devoted to the patients whom we all are here to serve. And that is not just data, or not big data in terms of size, but truly high-quality data that is curated, that is available for aggregation, and then readily presents itself in a sphere that's secure with regards to patient privacy, among other things, that presents itself in a sphere in which you will have all the tools that you may need in which to interrogate these now pristine data sets to address questions of relevance to you and ultimately to your patients. That is the objective. But remember that I said at the outset, it is derived from you. So as we go through these few slides in the next 15 minutes, I ask you to ask yourselves how you might be able to participate or encourage the participation. You'll see some who have already volunteered, not only in supporting Project DataSphere, but most importantly, by being providers of data sets. And those of you who have those, think of a very good reason why you shouldn't be a data provider, and then see me afterwards if you think you have a good reason. Because my job will be to convince you that that's not good enough. Those of you who don't have data sets, you have a voice. And I'm looking at my good friend, a patient and a patient advocate. Please, this is an opportunity to amplify your voice because it has already been and shall be heard. The CEO Roundtable on Cancer was derived and started by the 41st President of the United States in 2001 when George H.W. Bush called two people sitting at that table. There's the president on the left. In the middle is Bob Ingram, who at that time was running Glaxo Wellcome before it became GSK with its merger to SmithKline Beecham, and a fellow to his right by the name of Marty Murphy. We were asked by the president to do something in a corporate environment that might in some way change the trajectory of cancer if education and implementation of creativity derived from corporate executives might be brought to bear. Think of it. There had not been, I don't believe at least, at that time, an entity that really invited corporate executives from a whole host of industries to come together, gather around, and find something on which they could be bold and venturesome. And that was the challenge that the president gave us. When Bob Ingram asked him, "Sir, it's a great honor to be asked to do this, but what is success? What does it look like? What are you asking us to do?" And the president responded by saying, "Bob, you run a company of about 100,000 people. You set the agenda. What I'd like you fellows to do is to be bold and venturesome. Put yourself, and yes, maybe even your companies, at risk on behalf of cancer patients." And so it was, a group then of American companies, now of multinational companies, representing diverse industries gathered around together in institutions and formed this nonprofit 501c3 as the US IRS code. Well, here are some of the originating companies, and many of you represent these companies in this room. And they were joined then soon thereafter by some nonprofits that included the National Cancer Institute, a number of universities, and so forth, the CDC, all of whom came together. And when we recognized that amidst all of these various industries, there were representatives leading the companies that actually were committed to the life sciences, this is what that actually looked like. All of the CEOs representing insurance carriers and big banks and hardware stores and goodness knows what all, they're not shown on this slide. This is a subset of the CEO Roundtable of only those who have the privilege of leading companies devoted to the life sciences. And so you would see, as you might expect, the heads of most of the big pharma, many top-tier biotech, CROs, and a very important computer company that I'll come back to in a moment called SAS. We then posed to this group, let's call them the Life Sciences Consortium, LSC, which was then founded in 2004. We posed the same mission statement that the President had given to us. Be bold, be venturesome, do something together that none of your companies, no matter how large, neither a Pfizer nor a Merck nor whomever could do something alone, that if they banded together on behalf of patients, they might be able to do together. And so it was that they not only did that, but a number of other interesting early experiments that I haven't the time to go into. But ultimately, some 4 years ago, a group struck out to do something truly bold and venturesome, and that was to create a place of open access for cancer data sharing. And that platform would be voluntary, it would be nonprofit, it would be broadly shared and integrated, it would enable analysis to go forward within this entity. It would have sophisticated analytical tools, free of any charge, for no at any time would there be any charge for any of this. The industry, as well as the National Cancer Institute, among other individuals, would provide comparator arms, originally from phase 3 trials, now also from phase 2 trials, and very soon from active arms of those trials will be then available. And that these de-identified patient databases with the dictionary and protocol and CRFs would be embedded in this sphere, containing these powerful tools, made available free of any charge to the world's researchers, and it would be welcoming. One would not have to write a lengthy, detailed research proposal or grant, as it were. It takes about 90 seconds to apply. I encourage you to go to projectdatasphere.org. I know your names. I'll find you in there. And try it. You will find that within 2 days, you will have the keys to the kingdom, and you'll be able to peruse and use Project Datasphere to your own liking. The public launch was then on April the 8th, 2014. And I'm going to now introduce you to some of the members of the executive committee, Victor Zhao, who is head of the National Academy of Medicine, and Stephen Friend, who just moved to Apple, who is the chairman now of Sage Bio-Networks, a galactic thinker. Dennis Gillings, Sir Dennis, retired as the executive chairman, who founded Quintiles, a large CRO, a fine medical oncologist who's physician in chief of the James Cancer Hospital at the Ohio State University, Rich Goldberg, that fellow. Charles Sanders, who's chairman emeritus of the Foundation for the NIH, that our colleague Gidon has just referred to in some of the work that the FNIH does on behalf of the FDA and the NIH in general, and also Elia Zerhouni, who runs global research and development for Sanofi. The chair of the co-chairs of the Project Datasphere, we have one, fortunately, with us, and that's Dr. Mace Rothenberg. He co-chairs Project Datasphere along with our colleague Greg Kurt from AstraZeneca. Let's now go also and make certain that although most of these data derived that you find now embedded in Project Datasphere have been provided, de-identified, and pristine by their providers, namely large pharmaceutical companies and some modest in size biotechs. But also along this entire trajectory these years, we have tried and been grateful to try to align ourselves with not just the regulators called FDA and EMA, but those wonderful researchers who are members of the FDA and EMA faculty and have been guided and encouraged by the FDA and by the EMA. And Francesco, I want to acknowledge once again how grateful we are to provide you this progress report, because you have put wind in our sails by encouraging us to actually pursue that which we have done. So here are a number of just the meetings to let you know, since 2012, a number of meetings with the FDA, the IOM, and a very important first meeting with the EMA that took place in February of 2015, when we were invited by Francesco and Hans-Georg to make our first formal visit to Canary Wharf. And then there were a number of subsequent meetings with the United States FDA, such that now we have every 6 months a meeting with the FDA on this topic in the oncology hematology branch, of course, of that agency. And the most recent one, that was symposium number 1, of which I referred to November of last year, and now May of this year was the most recent one of those, in which we also were joined not only by the NCI, but the White House. Some of you have heard about the Moonshot. Yesterday there was a Moonshot summit in Washington led by Vice President Biden. The Project Datasphere is the alignment of that part of the Moonshot that deals with, quote, big data. So we are fully aligned, that is, Project Datasphere is a part of the Moonshot and vice versa. And it was at that last meeting of the FDA, the NCI, and the White House where a new project actually was proposed. And so it is not just a progress report that each of these meetings has actually produced, but in this case, actually a new project, a proposed research program of new intermediate endpoints in prostate cancer that would be explored within Project Datasphere, provided by the data that is there supported. And there were 2 investigators, both of whom come from Memorial Sloan Kettering, Michael Morris and Howard Shear, that were at that meeting and are pledged now to actually pursue that project derived as a consequence, not only the meeting, but most importantly, the availability of these data. And so we arrive at today, the 30th of June. Be it known that it is not raining and it's not windy, and we're in here doing the work. Let's talk about some metrics over these 2 years. Commitment from industry, including the NCI, in particular their cooperative groups, have provided more than 30,000 patient lives worth of data from 14 different data providers. There have been over 1,200 researchers who have access 24/7 the data in Datasphere at any time, and more than 4,000 times data sets have been downloaded. What I'm trying to express here is that it's not just porous, it's cordial, it's welcoming. It intends to be used, and it is being used by researchers, physicians, and scientists around the world. In fact, it turned out that there are so many high-quality phase 3 comparator arm, where docetaxel is the backbone, trials inside Datasphere, that we were encouraged actually to try a crowdsourcing experiment. And so it was that last year we executed the first prostate cancer DREAM challenge in collaboration with the IBM DREAM team and Sage Bionetworks, Stephen Friend, who I acknowledged earlier. And it turned out to be not only, it was the 32nd crowdsourcing, or a DREAM challenge thus far in the world done, but it was not only the largest, with more than 500 participants who formed 58 aggregate teams derived in 21 countries, but there were more participants in that challenge than all other 31 DREAM challenges. The peer-reviewed publications are now being derived from that. There are 2 now in preparation in Nature, and we'll talk about a couple of those towards the end. Prostate cancer, the proof of principle for the Project Datasphere truly must be and should be, again, back to you. You who provide the data, you patients whose data it is. Now the proof of principle is to answer questions of significance enough to pass peer review muster, and we'll talk just briefly about some of those publications, remembering that the door to Project Datasphere was open but less than 2 years ago. The timeline of this bespeaks, I would suggest, the very fact that it's driven by chief executive officers of industries who are measured on their output and on their agenda. What I mean to say is they really work. So the concept was first proposed by Mace and by Greg, Dr. Rothenberg and Dr. Kurt, in 2012, and it was approved at that CEO roundtable. Thereafter was spent a year of lawyerly work, and I want to acknowledge Hogan Lovells, for indeed they have provided millions of dollars of pro bono counsel preparing the contracts by which your companies or your institutions provide Project Datasphere, your data, in a de-identified fashion. And there's one contract that you've all agreed to. I salute all of you and your companies who have done so. And also, as well as the legalities, the privacy, namely SaaS privacy. You'll see that continued then into the second year, where resources were added, resources being not only the analytical resources, but human resources. We began to really gear up. And then it was launched and made available to the world. And in that first year, 7 charter providers provided. Our target was to raise at least 25,000 patient lives worth of data, of this high quality data, individual patient level, every data point of every patient, high quality curated data. And in fact, within that first year, it was superseded. There was more than 25,000 patient lives were actually provided. We had a moment's celebration, only to then have Mace and Greg launch us into our second year, and that's the year in which we find ourselves right now. And the target is to grow that from 25,000 to 100,000. So the first year we tripled from 0 to 3. Well, we're going now tripling again, and you'll see on the x-axis on the right-hand side where we intend to be by the end of this calendar year at approximately or more than 100,000 patient lives. It's being derived from this. You'll see where base was our 25,000 lives in 2015. That was the goal that was exceeded. We now have pledges from the cooperative group, in particular the Alliance, to add an additional 18,000 lives from the NCI. We have, and this is the Alliance component, where there are some almost 17,000 lives and that are going to be de-identified by a recently received grant from the Robert Wood Johnson Foundation, and that will bring us up to that level. We have additional data sets from industry, from 13 data sets that have already been pledged and are being de-identified as we are here today. But then there's this. We have 32,000 patient lives that are not yet pledged. There must be in this room representatives from industry who will raise your paddle and say, we'll provide 5 new data sets, or whatever that number is. Please go back and see how many data sets you can provide. You who don't have the data sets, use your voice, my dear friend, and encourage them to do so. The power of access to patient-level data is being seen. Tito Fojo, originally from the National Cancer Institute for 30 years, recently moved to Columbia University, said, and I quote, "The Project Datasphere platform has allowed the development of new models to predict prostate cancer tumor burden over time. Research that will have real implications for the drug development process and that can be repeated for many other tumor types if the right volume of data is made available." And I will not read further quotes because fortunately most of you hopefully have this flyer, and I encourage you to read those other testimonials from people like Dan Sargent, all of you who probably know of him as, in the States at least, one of the deans of clinical data analyses. And very recently, a paper was just published in Nature, and I have a copy of it here, and it's referenced there, entitled "A Patient-Level Data Meta-Analysis of Standard of Care Treatments for 8 Prostate Cancer Clinical Trials." All of this was done within Project Datasphere. And the final stanza does merit perhaps reading, where they said, "Open clinical trial data offer many opportunities for the scientific community to independently verify published results, to evaluate new hypotheses, and conduct meta-analyses. This study illustrates how the use of patient-level clinical trial data, parentheses, derived from Project Datasphere, enables meta-analyses that can provide new insights into clinical outcomes of standard of care treatments and thus, once validated, has the potential to help optimize healthcare delivery, that which all the oarsmen thus far today have been addressing." There's the full manuscript. You'll be able to read it online free of any charge. Here then are unfortunately not readily readable, but these are the first of the peer-reviewed papers that have appeared in the first 18 months since Project Datasphere opened its doors, beginning in May of 2015, and there are a number of papers, including papers in Lancet Oncology and additional papers in Nature that are in press, and quite a number, or a reasonable number, of abstracts and posters have also been published and presented at major meetings in Europe and also abroad, namely the United States. Notice how I did that. Here are the leadership, and I want to acknowledge very importantly in the lower right-hand corner, Mace Rothenberg, who there has been a rare Friday when he has not conducted every Friday afternoon at 3:30 to 4 a teleconference amongst the leadership of Project Datasphere week in and week out that has kept not only this Viking ship going fast and forward, but forward in a direction that's always patient-oriented. And with that, I express to you on behalf of the CEO Roundtable, but most importantly on behalf of those patients whose data truly are the ones who we are the hold it as a chalice within that Project Datasphere. Someone once told me that data in clinical trials really are patients with their tears removed. I think it's in that sense that we all are here. These data, these sacred data, are their data for us to use, but we're trying to put it in a fashion which it'll be all the more useful to every one of you. It'll get better the bigger it gets, because I promise you we'll take good care of it for you. Thank you very much.
Okay, so thanks. Okay, so we can go on and have the discussion afterwards. So Rick Kaplan from the Medical Research Council, single arms within platform trials.
Thanks, Paolo and Francesco. I'm going to try to dodge the difficult question for clinicians and regulators about what do you do when a single-arm trial has already demonstrated what looks like a breakthrough effect. Others have discussed that point. I'm going to try to talk about whether there are some options for how you can preserve a lot of the advantages of a single-arm trial, but actually do a randomized trial. It would have to be something which is roughly as quick and efficient, and that's what I think there's a way to propose on the basis of this being very much a large-scale platform trial era. You all know all these things, the NCI match trial with 24 arms currently open, I think. In the UK, there's the lung matrix trial with about 20 arms open. The lung map trial in the US is similar. We have here in colorectal cancer something called the Focus 4 trial, which I'll talk about later. But quite an important element of that is, although most of it has to do with a series of stratified comparisons against controls or placebos, there is also a non-stratified cohort within that trial, which means effectively that this trial can pick up almost all of the eligible patients with advanced colorectal cancer in the UK. Each of these trials has a tremendous capacity, and in this sort of context, even something like ROS1 at 1.5% of lung cancer patients represents a situation which is not genuinely rare. It offers plenty of opportunity on a national or an international basis to do larger scale testing. Whether that applies as well to true rare diseases or not, I'm not sure, and that we can talk about that more. So we've talked a lot today about objective response and how helpful it is. Certainly it is helpful to developers and exciting to all sorts of parties that are relevant here. We've talked about some of the difficulties with RECIST measurements and pseudoprogression and how you see some changes in waterfall plots even for placebo arms. We haven't talked very much about the fact that combinations are really a minefield when it comes to looking at objective response rate or even duration. Segregating the differences in combinations from those you might have seen with single active agents in the same setting is particularly difficult without a control group. Moreover, proof of benefit almost always seems to require either a time to event endpoint or, in the context of a lot of today's discussion, duration endpoint, still a time-based endpoint. The drawbacks, again, we've talked about before, the historical data comparisons, the impact of pretreatment parameters, which we may or may not be able to identify, that are usually greater than the impact of most treatments that aren't breakthrough treatments. And we may indeed be able to minimize the effect of these things by extensive characterization of large databases of patients, as you've just heard. But for many agents and settings, objective response or duration is not going to be all that is needed in terms of important outcomes. And indeed, they may be helpful in terms of providing a go decision for something, but not very reliable in providing a no-go decision. And we've talked, everybody else has talked about the fact that separating prognostic and predictive effects is challenging there. Dan Sargent, whose name you just happened to hear, has done some modeling work that shows the variability in historical control success rates, outcome drifts, and patient populations over time, and how the false positive and false negative error rates in single-arm designs are 2 to 4 times, the false positives are 2 to 4 times higher than in randomized phase 2 trials consistently across modeling that's been done. And similar work has been done by others in the US and the UK that demonstrates the same point. Increasing sample size per se is not enough to overcorrect the optimism of single-arm trials. Again, these are points that you've heard before, but not perhaps the emphasis on combinations. And indeed, even as we come through some major breakthroughs, and even as we have the Nivos and Pembros of this world, combinations are going to be increasingly important for the next step forward, and that much is already clear. So that the need for designs that can accommodate those situations as well is quite important. Also, objective response rate is, in practical terms, limited to the neoadjuvant setting and the highly resistant sort of end-stage disease setting, where we may well be able to make a stab at understanding what the results mean. But anywhere in between, it's very difficult. And we would, of course, like to investigate new agents at earlier stages in the disease natural history than the end stage. We've talked very much about the fact that apparently very promising results can make subsequent randomized trials more difficult. And most importantly, it might be time inefficient, or at least neutral, to do single-arm trials when you have a large platform trial in which you can ask the question. So I'll go back to this colorectal cancer trial design for a second, and just picture any one of those cohorts where we'll be randomizing potentially between a novel agent and a placebo. This happens to be a maintenance trial setting. And that's why placebos are are in this particular, but the same applies if we were talking about a different setting and an active control. Companies that we work with very often say, well, we'd like to just go through the first stage just as single arm and see if we get a signal, because that's going to give us a signal ahead of anything else. This trial has been set up to be a rolling phase 2-3 trial, and we've tried to make sure that we randomize from the beginning for the efficiencies that adds. Now, I don't think you'll be able to see very much of this, but this is a time scale of about 16 months for one of the comparisons within the Focus 4 cohort. This was on a cohort that's about 20% of all comers, and we had proposed a randomized design against placebo with a 2-to-1 randomization, looking first at objective response and then on at PFS, which is the main endpoint. And the 16-month, the end of that chart, is where we would say we would be getting our randomized phase 2 result, looking for a target hazard ratio of 0.5. What the company wanted to do was do an analysis. This looks like 3 or 4 months of registration of patients, and then several months of waiting to see how many objective responses we got, and then reporting that to the company. That turns out to give these figures. If we did this as a single-arm phase 2 with this size cohort, there'd be 27 patients recruited over 3 months. It would take 16 weeks for the full assessment of response, 1 month to go over the data checking analysis, and in 7 months we'd be able to say whether we were getting a positive signal on objective response alone, a single arm. If that were encouraging, however, we need to go and set up a randomized phase 2 trial unless this is a breakthrough. And the subtotal, just to get that second part started, takes us up to about 13 months from the point we started, and the total elapsed time, or the total time up to that point, is 20 to 30 months before a phase 2 is completed. The comparison in the same setting with the same number of patients coming into the trial at any point, if you looked at 9 months, you would have 24 patients on the active arm evaluable for response, plus you'd have the stage 1 progression-free survival analysis available. There would have been a total of 81 patients at that point. This is 2 months later than they would have got the uncontrolled initial results. And at 16 months, as compared to 24 or 30, we would have the final phase 2 DFS analysis as well. That was convincing to the company involved, and that's the direction the trial can proceed in this case. So there's several arguments that are worth thinking about here. You potentially can provide, in many settings, randomized phase 2 data to give no-go decisions just about as quickly as you could in a single arm. The go decisions become go-on decisions with the next needed data set already underway. I mean, you could say, yes, that's the expansion cohort equivalent that's underway, but the point is that there is randomized control here. There isn't a disadvantage if the response rate, the response data are so dramatic that they are ready to approach regulators with in the context that we've been talking about today. In the meantime, if that's not the case, you provide a much fuller toxicity and safety profile, pharmacodynamics and potential for HTA assessment and so forth, plus presumably relevant biomarker data and translational data that may be relevant, and the time frame could be very similar. Again, it's not going to work for chordomas, I presume, unless we can have an international platform for chordomas, but it may work for very many of the other diseases that we're talking about today. And the more the durable responses are critical to useful data, then the better the argument is for randomizing and a seamless design right from the beginning. And I'll stop there.
Jonas Berg, at the Karolinska. I must say, MRC is, at least from my viewpoint, I'm now not talking as the acting chair for SAG Oncology, Hematology here in London, but from a researcher's point of view at Karolinska, I think this is a much better setup with this type of multiple randomization based on relatively well-established biomarker-defined subtypes with a common entity. The only concern from my viewpoint is that the more we learn about at least some solid tumors and look at the heterogeneity within the tumors, and we also look at what happened during tumor progression, that some mutation, alteration of pathway may emerge during tumor progression. So I like your model in principle with present knowledge, absolutely. But the problem is, how do you build into this model the dynamics? I saw to my pleasure, although, that you had a biopsy at relapse. And that is, of course, if you even can build into that system, for example, if you have you were stratified not to have a RAF mutation, and then suddenly a progression in other arm, you develop a RAF mutation in the progressed disease, then you have an algorithm for that strategy too. Then I think you are much better off than single-arm study, and I'm fully supportive that a randomized phase 2 design in principle is always, at least from my viewpoint, much better than a single-arm study in all dimensions. Although the statistics is, of course, is an issue there, but from my biological and comparative point of view, just to be congratulated to this type of strategy.
Thank you. I think that that's a very key element, and it has been a key element in the design of that particular trial from the beginning, a recognition that we may start down this stream and have to branch off at various points and reset our sample size and go on. But again, once you're in the context of a big umbrella trial, there is a way forward, and it is potentially practical. And actually, Sabine Tejpar, on your right, who's it's been key in helping us try to navigate that particular minefield. We're still trying to model how much it slows us down as we move forward, but I think there's no disadvantage in this frame. This framework is as good as anything else for handling that very difficult problem.
So my name is Lena Arfeld. I'm representing a small biotech company, and I just want to comment on your at the advantage of the randomized trials. So I think in the companies we have a lot of interesting compounds for development, and by approaching the single-arm trials compared to the randomized, it's also about the amount of the investment that the company is doing, because the cost of doing the larger trials earlier is of course much higher than to do the single-arm trials, where you can have an early efficacy read before you have the large investment up. So that's definitely an important component in particular for the SME companies.
Totally understood, and there is that tension that time is also money. This is reasonable in terms of time. It may not be any more costly in terms of time. It certainly could be more costly in terms of the actual drug costs for a very small company. As a practical matter, the things that are getting to phase 2, for the most part, you fairly quickly develop some partnerships that make it possible with larger companies that make that possible. But I take your point.
Maybe I have a question, Dr. Kaplan. What is your experience in terms of this type of trials with multiple arms and sometimes multiple companies? Is this an attractive, let's say, business model in practice? Is it working, or is there still some reluctance for this type of cooperations?
I don't think there's a lot of reluctance. I think that until something comes out of a model like this that is a clear and obvious success, it's going to be a difficult decision for many companies to commit in that direction. But I think the competition is tough. There are lots of drugs in development and not as many niches for drugs, and companies need to look for places where they can keep their possibilities open rather than prune them all off to the one or two trials they can focus on. So I think the environment is probably right. And certainly, since we're not comparing one drug, in this particular model, we're not comparing one drug to another at all, there shouldn't be a disadvantage for companies. But again, we need to see this all succeed.
I would like to ask Dr. Blumenthal, if you have a breakthrough agent with a single-arm study and it is approved, and the next company comes with a Me-Too agent, how do you improve it? Is a single-arm study enough? Will you require a randomized trial? How are you dealing with the situation?
Yeah, good question. So for agents that are approved under accelerated approval, they're not considered available therapy. So if you have a drug that's approved under accelerated approval, the next drug with a similar mechanism of action could also get approval based on a single-arm trial until that drug, that first drug, if you will, converts to a full approval. Then all other companies are sort of blocked from the accelerated approval mechanism. If it's if it's a traditional approval based on a single-arm trial, such as was seen with the hedgehog inhibitors and basal cell carcinoma, vismodegib was approved, and then I forget the name of it, but there was a subsequent approval, Novartis drug, also approved traditional approval based on single-arm trial.
From my point of view as a patient, with the health organisation finding hard to fund new drugs, how can we absorb this into an area where inequality throughout Europe is stopping access to the current drugs? So how can the new drugs actually get out to the people that need it?
No one dares to answer. My comment to this is, of course, this is a political matter of priorities among the politicians in the different European countries. EMA is perfectly well serving drug approvals. Then is it up to each country to make HTA evaluation? And human life is much less worth in United Kingdom compared with other countries. So that is a matter for your politician. That's why it's extra bad that you leave the European Union.
This is a question to Dr. Blumenthal. For those drugs being approved on a single-arm objective response rate, does FDA asking post-marketing commitment? If yes, what is the policy of the data that you want to have afterwards, just to confirm or not the initial decision?
Yeah, so usually for an accelerated approval, typically we ask for a randomized controlled trial as confirmatory evidence. Now, there can be issues where once it's out on the market, it's hard to randomize in the same setting. So we've accepted randomized control studies in, say, earlier lines of therapy, or even in some cases different but related diseases for confirmatory evidence. For very ultra-rare populations, there may be a mechanism For example, if you approve based on a single-arm trial, perhaps longer duration of follow-up as the confirmatory evidence, or as we move more into real-world settings where the EMR data becomes more robust, perhaps just speculating down the road, maybe the real-world data could potentially serve as confirmatory evidence.
Yeah, I have a different question for Dr. Murphy. You laid out a very nice case for how Project DataSphere evolved from its initial conception to where you are today. And my question is, how would you see the most immediate way that the information that's already there can be used to inform the challenges we're discussing today and the opportunities around the use of single-arm studies for some of the decision-making in oncology specifically?
Thanks very much for that question, that opportunity. I would give you an example of one, but it's only an example. I think there are many others, and I'm sure the other people here would think of additional ones as well. But with regards to, let's say, the prostate cancer comparator arm of phase 3 trials that now I think in Project DataSphere is the world's largest repository of those curated, de-identified trials. There are, I believe, right now 11 phase 3s. And what has taken place is that 8 of those now have been aggregated. Remember, they're all curated. They've now been aggregated, and you have more than just the sum of the parts. You have essentially a new data set, haven't you? And now this new data set has been uploaded back into Project DataSphere. And the message, this is very recent, and the message will soon go out amongst all the users of Project DataSphere, will be that this new data set consisting of the size and complexity that it will be defined as is now available for your new study. One of the things, and I would ask actually Guidon to comment on this, and that is some of our interactions with the FDA as an agency has been encouraging us to look at the rare tumors. Pediatric is an example that we're now seeking, aren't we, Mace? And also other rare cancers in the hopes that perhaps in aggregate of pristine data that is properly curated and then uploaded, such as has now taken place, if you will, archetypally in prostate cancer, but in now rarer tumors, might it not serve for agencies to use as truly a robust, if it's large enough, robust comparator arm that might therefore provide everyone succor and surcease with regards to can you now do a single-arm trial without as much hesitation as we've been talking about today? Mace, may I turn it to you and then to Guidon?
A couple of things that come to mind, Ramsey. One is, we've been talking about single-arm trial data being used as a potential basis for medical authorization here. That's really restricted to the category of trials that have extraordinarily positive data. There are a whole more frequent category where the data look good, but maybe not as good as to be sufficient for registration. Then, as sponsor, we face a very difficult decision. How rigorous is this phase 2? Do we do another phase 2? Do we go into phase 3? And as we've all seen who've been in this field long enough, we've seen the great results from phase 2 that fail in phase 3. So what could we do to increase the likelihood that what we're seeing is a true drug effect and not just due to patient selection? That's one potential use of the data from Project DataSphere. You could potentially, based on the clinical data that's already there, do basically a matched control with hundreds or thousands of other patients that can give you a very clear idea as to whether or not what you're seeing is due to the intervention or just due to good patient selection. Second part of this is actually a forward-looking statement, if I might, and that is one of the criticisms that's been leveled at Project DataSphere is that you only include control arm data. That's now out of date. It may not be as relevant because many of these were from positive phase 3 trials that show the investigational arm has become the standard of care. And we realized that, but we felt it was important for us to be able to walk before we could run, and that there would be a number of very valuable analyses that could be done off of this database. But clearly our goal, and in the not too distant future, is now to raise the stakes, to say, with the experience that we've had of the data integrity that's been demonstrated, to actually now ask sponsors, companies, cooperative groups, large cancer centers, to upload both the investigational arm and the control arm. Now, for some of us, that made us shudder with the idea that someone could go in to the data that we've used, let's say to base a regulatory filing, reanalyze the data using some different techniques and come up with a different result. That could cause mass confusion. So actually, it was a colleague who's part of Project DataSphere from Amgen who came up with a very simple, elegant solution. Said, rather than uploading 100% of the data, upload 85%. In that way, no investigator could recreate the clinical trial and have the results vary or be consistent with the same level of consistency as the original trial. So we think that that's going to be the next step, and we'll be, again, approaching many of you with requests for those data, and I think that that's going to make it even more robust. But to bring it back to the presentations today, there's a very nice publication, presentation by Gidon Blumenthal about looking at intermediate endpoints in predicting ultimate outcomes, overall survival, for instance, on intermediate endpoints from targeted agents or from immunotherapy. This is an area that's extremely relevant, extremely urgent that we get right, because as we develop agents that have better and better effect, lead to longer and longer survival, become more available, which allow patients to cross over, we're faced with the challenge of how to accurately and reliably evaluate new agents when other effective agents are available down the line that could obscure the impact on distant endpoints like long-term overall survival. So actually, this applies to immuno-oncology and checkpoint inhibitors, and this might be an opportunity for those companies that are involved in checkpoint inhibitor development to actually upload their data, this is all new, all current data, to then allow pooling of that data to actually address issues that could not be identified in the analysis that Gidon and his colleagues did on the data sets that were available to FDA, but this would actually broaden it. It would put it in a neutral territory, a sort of Switzerland of cancer research, known as Project DataSphere, and then allow all the companies access to this de-identified patient-level data, and not just cancer investigators. This is open to people who are in computational biology from centers that you never even heard of, come up with these new algorithms that can allow us to actually look at early intermediate endpoints that reliably predict long-term outcomes and can speed the development of new drugs, which is what we're all hoping to see. So I know it's a long-winded answer, and I went off in several different directions, but I think this is such a critically important area. Gidon?
Yeah, thanks, May. So I think, well, two thoughts. One, I think the idea of sort of liberating the data. I mean, we at FDA are sort of, we have this whole treasure trove of data that we have access to and we can do analyses on. But I think the real value of endeavors like Project DataSphere is to, have sort of outside the box thinkers from different, engineering backgrounds, physics backgrounds, what have you, computational sciences, also interrogate the data from a completely different standpoint. So that would be one value. With respect to sort of match controls and rare subsets, even in lung cancer, where we have all these tens of thousands of patients in randomized controlled trials, I think it's going to be difficult. It's going to take a lot of conversation with statisticians, epidemiologists to sort of nail down the methodologies that would make everyone comfortable for regulatory approval. And then I think the problem becomes even more magnified in rare subsets where you might be making assumptions on even smaller patient populations. So I think it's something definitely to look at, but I think it's going to be challenging.
I do think the last wording, it will be challenging. I have no problem that one is doing this type of exercise with different databases. The only thing is that those databases really need to be much more fine-tuned and detailed into the minute details, because we started this day by discussing specific molecular targets and how dramatically more pronounced the effect in some very well-defined subgroups. The problem is that most of the agents today are developed versus sophisticated targets, and I think one of the problems with present development is that we are investing too little in really trying to understand the biology. And then some people will say to me, oh, that has been so far a failure. But I would say these databases, without the detailed molecular characterization, will be of, I would say, less pronounced value. And once more underline, metastatic disease is not the same as primary disease. We really also have to have that insight when we are trying to understand why are drugs working, not working. And that database, which we have been discussing, would be tremendously more important if we have detailed molecular characterization of each individual. Then we may try to improve, because computerized play, because if there is no biological rationale for a finding, then I would be concerned just by computerized algorithm that you have a quote-unquote chance finding. That would be very dangerous.
Here, here. The qualification I should have provided but didn't, is that, as May said, that now very soon active arm data is going to be not just only allowed but encouraged, but also in the entire project data sphere, the availability of genomics, proteomics, all of the omics is already designed, and it is going to be available for uploading. The challenge, and I put it to all of us, is the term de-identification. Because the more, as we know, you include genomics into a patient's record, the greater the probability or possibility, I should say, that record, if one tried, could be re-identified. So the point here is more of a legal issue, and it's not so much any longer, thankfully, because of SAS and a lot of other technologies, a technological issue, but it's a legal one. But as long as it's legal, and as long as our attorneys provide pro bono time, we will come up with solutions, I can assure you.